Okay, so ladies and gentlemen, good morning. My name is Mukhtar Aliyu. In today's video, we are going to look at the transfer RNA, which is one of the types of RNA molecules that we have in the cell. So, what is transfer RNA? Transfer RNA is a type of RNA molecules that plays a crucial role in protein synthesis. So, remember, in a cell, we have DNA converted into messenger RNA. Then the messenger RNA transports itself from the nucleus, that is where it is synthesized, to the cytoplasm for protein synthesis. So a transfer RNA is also another type of RNA molecules that use in protein synthesis. And it serves as an adapter that translates genetic code from mRNA into a specific sequence of amino acids. So the genetic code, there are triplet codons that are also found in the cells, which use to translate the information in mRNA molecules using transfer RNA as an adapter molecule. So we are going to see how the anticodons of a transfer RNA will be used to use a codons from the messenger RNA for protein synthesis. So understanding transfer RNA is essential for how proteins are constructed in living organisms. So you need to understand the transfer RNA, and that will give you an idea of how proteins are constructed in the body of living organisms. So this is actually, although this is a ribosome is a part of a cell where protein synthesis takes place. And these are the different types of transfer RNA. These are different types of transfer RNA. This is a transfer RNA. This is also another transfer RNA. This is also another transfer RNA. And you can see that clearly this transfer RNA is responsible for carrying amino acids. So this is one of the amino acid carried by this transfer RNA. And this is also another transfer RNA that carry another amino acid which used for the synthesis of proteins. So you see this one is also amino acid started forming the peptide bond. So we have 3 amino acid here, but each transfer RNA is responsible for carrying one amino acid, and we'll see that. So what are the structure of transfer RNA? So transfer RNA has a distinctive clover leaf structure. It has leaf structure that is critical for its function. So it has a distinctive clover leaf structure. So if you ask, what is the shape of the transfer RNA? So it has a clover leaf structure, and it consists of 3 main loops, including the anticodon loop that recognize mRNA codons. And then it also contains the 3 end of transfer RNA, which carries an amino acid linking it to the protein synthesis process. So let's see the transfer RNA. So you can see this is an anticodon loop where it actually has anticodons, and each of these anticodons is complementary to a sequence of codons in the transfer RNA. So that is why it is these anticodons that use to recognize the mRNA codons. So you see the anticodons here, CUC, will recognize codons from messenger RNA, which is GAG. So this is how you use a transfer RNA where you have an anticodons, and this anticodons is responsible for recognizing a codons from the messenger RNA. And very importantly, there is a 3' end of the transfer RNA, and the major functions of the 3' end of transfer RNA is to carry a specific amino acid. So this is the point of amino acid attachment. This is the point where amino acid attach itself, and this amino acid is used to link the growing amino acid chain for protein synthesis process. we also have T loop, we also have D loop. So these are the three loops that are found in the transfer RNA. We have T loop, we have D loop, and we have anticodons loop. So functions and translation. Transfer RNA is responsible for bringing amino acids to the ribosomes during translation. So it is actually the work of transfer RNA to bring an amino acid to the ribosome for protein synthesis. And each transfer RNA is specific to one amino acid, ensuring the correct building blocks are used. So you see, we have 20 amino acids that are found in living organism, and those 20 amino acids has their own specific transfer RNA. So there are 20 amino acids, and each of these amino acids has their own specific transfer RNA molecules, ensuring that there is correct building blocks, that is amino acid, is used in the protein synthesis, and we'll see that. So the interaction between transfer RNA and mRNA codons ensure that the genetic code is accurately translated into protein. So let's see. You see, this is actually a transfer RNA with its anticodons, and the anticodons here is CGU, and it carries an amino acid alanine. So you can see that it carries amino acid alanine at the 3' end. And don't forget that I told you that this transfer RNA is used in, so this 3' end of transfer RNA is used for amino acids attachment. So you see here you have alanine attached itself here, and then you have anticodons loop which are used. Look at it here. You can see that you have an anticodons here that are used to recognize the codons from the from the mRNA. So you see here we have alanine. So this transfer RNA carrying alanine, it's now recognize the codons from the messenger RNA, and then the amino acid is now linked to the growing polypeptide chain. Then the next moves to next transfer RNA. So you see, as the amino acids is attached to the growing chain, then the transfer RNA will detach itself from the messenger RNA to go back and carry another amino acid. Then it will continue like this. So as you add, more of the transfer RNA will be attaching themselves to the codons from the messenger RNA, which will actually deposit the amino acid in the growing polypeptide chain and then go back to carry the same amino acid. So it will keep recycling itself. This is how proteins are synthesized. So we have different transfer RNA. Each of these transfer RNA, they are responsible for transporting a specific types of amino acids. As they deposit the amino acids, then another transfer RNA will now attach itself, deposit another amino acid, and then it will detach itself. Then another one will now bind to the codons in the messenger RNA in the ribosomes, and this is how it continues until the proteins is fully synthesized. So this is actually the functions of transfer RNA in translation. It carries amino acids, attach it to the growing chain to detach itself, then another one recognize another codons in the messenger RNA, deposit the amino acids, then detach itself. So this is how it continues till proteins is fully synthesized in the body. So then we have aminoacyl transfer RNA synthetase. There are enzymes that are actually attach the correct amino acid to its corresponding transfer RNA. So each transfer RNA has its own specific aminoacyl transfer RNA synthetase enzymes, because this enzyme is responsible for attaching a specific amino acid to the corresponding transfer RNA. And this process is critical for maintaining the fidelity of protein synthesis, because if amino acid it wrongly attach itself not to its corresponding transfer RNA, then there is a likelihood there will be a problem and there will be no fidelity. So, but because of these different enzymes that are found which are specific to each transfer RNA, so it are different amino acids will be attached to its corresponding transfer RNA and used for protein synthesis. So there are 20 different synthetases. There are 20 different synthetase enzymes, one for each amino acid, highlighting the specificity of this system. So this is what actually happened. You see, this is where we have tyrosine transfer RNA synthetase. So we can have alanine transfer RNA synthetases. So you see this tyrosine transfer RNA synthetases is actually the enzymes that use for attaching tyrosine amino acid to its corresponding transfer RNA. So this is tyrosine transfer RNA. It means that it is transfer RNA carrying tyrosine amino acid. And in the process of this attaching the amino acids to the transfer RNA, you can see clearly here, this is the transfer RNA. This is its active site for the transfer RNA, and this is the active site for the tyrosine. So you see the transfer RNA and the tyrosine, they all actually bind to the active site, and after the bind, then the catalytic synthesis takes place. And you see here there is a covalent bond formation between the tyrosine and the 3' end of the transfer RNA, and here you have a fully synthesized, or you have a fully charged tyrosine transfer RNA. So this is what happens. So you see the transfer RNA and it is cognate amino acid enter the active site of the specific synthetase. So you see they are now enter the active site here. So after they enter the active site, then the next thing, using ATP. So ATP, it utilizes synthetases, catalyzes the covalent bonding between the amino acid and the transfer RNA. So in these reactions, for these reactions to take place, the covalent bond formation between the amino acid and the transfer RNA, it consumes ATP. So ATP is consumed. So after the reaction is completed, then you will have your charged transfer RNA, and then you will have your free enzyme. So the transfer RNA charged with the amino acid is released by the synthetase enzymes. So this is how aminoacyl transfer RNA synthetases charged transfer RNA for translation process. So what is the role of anticodons? So the anticodons is a sequence of 3 nucleotides on transfer RNA that pairs with the codon on mRNA. So remember, we have anticodons loop, and this anticodons loop usually contain 3 nucleotides. And these 3 nucleotides are responsible for pairing with the codons on the messenger RNA, which actually leads to the deposition of the amino acid carried by that transfer RNA. So this complementary base pairing ensures that the correct amino acid is incorporated into the growing polypeptide chain. So because anticodons is complementary to the messenger RNA codons. So it ensures that that complementary base pairing is maintained for polypeptide chain synthesis fidelity. So the specificity of anticodon-codon interaction is crucial for accuracy of protein synthesis. You see, this is what you have. So this is a transfer RNA carrying methionine, and this is the codon from the messenger RNA, and these are the anticodons from the transfer RNA. So they will recognize each other. So after this recognition and base pairing, then the amino acid will be deposited and used in the polypeptide chain synthesis. So transfer RNA in different organisms. Transfer RNA molecules are found in all organisms from bacteria to humans, while the basic structure and functions of transfer RNA are conserved. conserved. There are variation across species. So, but there are some part of the transfer RNA that can vary across one species to another. But the functions and the structure of the transfer RNA are conserved across all organisms. So understanding these differences can provide insight into evolutionary relationships and cellular function. So now the next thing is transfer RNA and genetic code. The genetic code consists of 64 codons. So there are 64 codons for this genetic code, which corresponds to the 20 amino acids used in proteins. So transfer RNA molecules have reached the gap between codon in mRNA and amino acids they encode. So this precise translation process is essential for proper expression of the genes. So this is actually, ladies and gentlemen, the genetic codes. We have 64 of them. We have stop codons and we have the codons that are responsible for coding an amino acids. So what are those stop codons? So you can see here, ladies and gentlemen, you have A, here is A. Okay, let me start from here. You have T, A, G is a stop codon. You have T, you have this T here. Then you now have, let me actually teach you how to read this. You have A here, then you have A here, then you have G. But some of you will now see the stop codons are U, A, U, A, G, and then U, G, A. But why are you seeing T? But remember, ladies and gentlemen, T is replaced with U. So you see here, you can replace it as U, A, G. That is U are gone. So you see, T is replaced with U. So it means that you are reading from here TAG. So TAG is a stop codon, or you can say UAG, or you can say UAA. You have A here, you have A here. So you have UAA or TAA is a stop codon. You also have another stop codon here, TGA or UGA. So T is replaced with U. So now, I can decide to ask you, or you can decide to ask, like, throwing in here, what are the possible codons of threonine here? So threonine has many codons, because one thing with anticodons or with genetic code, more than one genetic code can code for one amino acid. You can have 5 genetic codes that are coding for the same amino acids. Like for example, you can see now here A, C, T here, it is coding for threonine. At the same time, A, C, C, threonine. A, C, E, threonine. A, C, G, threonine. So that's what happened to genetic code. So the genetic code in the mRNA and the codons in the messenger RNA are used to code for a specific amino acids, which used in the translation. So the precise translation process is essential for proper expression of agents. So ladies and gentlemen, these are the genetic codes that are responsible for coding for different types of amino acids. So transfer RNA, it helps bridge the gap between the genetic codes in mRNA and the amino acids they encode. So this is what we call genetic code. Although I'm going to do also a video on genetic codes and you will enjoy it. So this is just to show you the role of the transfer RNA in genetic code and protein synthesis. So summary and importance. So transfer RNA is vital for translating genetics information into functional proteins. So that is one of the functions and importance of the transfer RNA. Its unique structure and specific interactions ensure the accuracy. So the interactions between the transfer RNA and the messenger RNA, it ensures the accuracy of protein synthesis. So research on transfer RNA continues to reveal important insight into cellular functions, genetics, and biotechnology. So ladies and gentlemen, don't forget to like, share, and join my YouTube community. So in the next video, we are going to talk about the ribosomal RNA.